The adaptive immune system can be and is a very complicated system to understand. So, what we're going to try to do is really highlight the main players that are involved. For example, your lymphatic organs and what they do can be divided into what's called primary versus secondary lymphatic organs. We're going to make sure we understand the difference between the lymphocytes. We're going to focus on three of them: the cytotoxic T cells, the helper T cells, and the B cells. And we're going to understand them in the context of two types of adaptive immunity. One is known as the cell-mediated immunity, and the other known as the antibody-mediated immunity. If you think of a pathogen, a virus or a bacterium, they're going to be in one of two places in your body. They're either going to be inside of your cells, like a virus, or they're going to be outside of the cells, like some viruses can be floating around outside of cells or bacteria. So you have to have two ways to get to them. The cell-mediated immune system is more actively looking for intracellular pathogens. So it's very good at finding cells, your own cells, that are infected with viruses. Antibodies are much better at finding pathogens that are floating around in the extracellular fluids. So we have two tiers when it comes to the adaptive system, two levels: the cell-mediated and the antibody-mediated. So let's work on this figure. The top, we see where lymphocytes come from. They all come from bone marrow. So bone marrow is a primary lymphatic organ. It's the location of the production of lymphocytes. Both T lymphocytes and B lymphocytes come from bone marrow. B lymphocytes are ready to go. So when B lymphocytes leave your bone marrow, they are mature. They're ready to go. Where do they go? They go to what's called secondary lymphatic organs and tissues. Those include lymph nodes, the spleen, tonsils, and other lymphatic tissues. So secondary lymphatic tissues are where lymphocytes go after they're mature and basically wait. And they wait for the day that they can fight an infectious organism. So this green thing represents a secondary lymphatic organ or tissue, like a lymph node. T cells are a little different. Before they're mature, they have to go to the thymus for further training. So T cells that leave bone marrow are known as pre-T cells. They're not quite ready yet. And there's a number of mechanisms in the thymus that allow those pre-T cells, kind of like a pre-teenager, to mature. And once mature, they get to leave the thymus and go to those same secondary lymphatic organs that the B cells went to, like lymph nodes, like the spleen, like tonsils. So T comes from thymus. T cells have to go to the thymus. And the thymus is kind of like another training ground for them to learn how to be T cells. All right, so now we're in this secondary lymphatic organ, this lymph node. And then it all depends on whether these lymphocytes can detect a pathogen or not. And how they detect pathogens is they have antigen receptors. So the B cell has these antigen receptors that can detect a bacterium or a virus. The T cells have these antigen receptors on their surface. Notice we've got helper T cells and cytotoxic T cells. Let's look at the helper T cells. The name kind of tells you what they do. They're going to help everyone. They don't just help cytotoxic T cells. They also help B cells. What we mean by help is to amplify or help to activate these lymphocytes. They help to boost your immune system. You would have an immune system if you didn't have helpers, but it would be very, very weak. We need it to be very, very strong and robust. So the helper T cells actually produce chemicals that boost your immune system. If you think about HIV is a virus that destroys the helpers. And if you destroy the helpers, your entire immune system collapses and you don't have a strong immune system. So individuals with HIV or AIDS, the immune system is so compromised that they often get infections that normally wouldn't be there if you had an immune system. We call those opportunistic infections. So helper T cells help. You'll notice another name for a helper T cell is a CD4 positive cell. They have what's called the CD4 receptor, but we'll talk more about what that does another slide. Cytotoxic T cells tell you what they do. They are toxic to cells. They can destroy cells. The cells that they're most accustomed to destroying are cells that are infected with viruses. So if you have the flu, and that flu virus is inside of a respiratory cell, a cytotoxic T cell will destroy that respiratory cell. But that way we can destroy the viruses. So it's like destroying the country that's harboring the enemies. If you're harboring an enemy and letting that enemy survive and expand, you're just as guilty. So we have to destroy the cells that are infected with viruses. And that is described down here. And this is called cell-mediated immunity. It's directed against intracellular pathogens, which are viruses. It can also kill cancer cells, and unfortunately, it'll destroy tissue transplants. So we have to suppress these when someone has a tissue transplant. B cells do things a little differently. When they come across the pathogen, they produce antibodies. And when a B cell is producing antibodies, we call it a plasma cell or a plasma B cell. And antibodies are a little different. Antibodies don't get into cells. They attack antigens or pathogens in fluids. So a fluid, another name for a fluid is a humor, H-U-M-O-R. So antibody-mediated immunity is also known as humoral immunity because antibodies go into your body fluids and look for pathogens there. So there's a lot on this figure, and it's an introductory figure, but you know it's pretty complicated. What I recommend doing is one day go back to this figure because it might not make sense now, but by the time we get to the end of this PowerPoint, we should be able to go back to this figure and have it make a lot of sense. Notice there's a lot of proliferation occurring. Proliferation means cell division. So now if this is a single cytotoxic T cell, look what it did. It proliferated into a bunch of them that are actively going out there looking for pathogens and memory cells. These memory cells, memory cytotoxic, memory helper, memory B cells, are the reason why your adaptive system adapts or gets better. It's because every time you run into a pathogen, your immune system leaves behind memory cells. And those memory cells will fight that infection much stronger because it has more cells waiting. That's also the basis of a vaccination. A vaccination, the whole goal of vaccinating is to produce memory lymphocytes. Let's quickly go over some of these organs like the thymus. We know where it is. It's in our chest. It sits above our heart. So it's located in the mediastinum. It's a bilobed organ. It's a primary lymphatic organ for the maturation or the education of pre-T cells. Trains those T cells on how to be a T cell. T cells that can't pass the T cell test have to be destroyed and removed. So there's dendritic cells and macrophages that remove T cells that don't make the cut. So really, most suggest that only about 2% of the T cells survive the thymus. It's a very rigorous program. Only the top of the class get to leave the thymus and become a functional, mature T cell. This way, your immune system doesn't make any mistakes. We don't need a T cell out there that doesn't quite know how to fight an infectious organism. So it's a very rigorous program in the thymus. You'll notice that this drawing is the thymus of an adolescent. As we age, the thymus gets smaller and smaller and smaller. So the thymus is much bigger when we're a child and an adolescent because it's training more. We're getting exposed to pathogens early in life and training our immune system. As we age, our immune system doesn't respond as well. In elderly individuals, they get sick more often because they don't have that immune system's ability to train new T cells. So that means early in life we want to be exposed. We want to expose our immune system to organisms so it can learn, so it can train while we have a nice functional thymus. So let children play in dirt. That's the whole basis of that. When you let children experience the environment, they get exposed to things. Obviously, you don't want to expose your children to tuberculosis, but you want to expose children to not so pathogenic microbes so you can train the immune system. Lymph nodes scattered throughout the body, over 600 of them, different locations, different regions. Lymph flows into them using afferent lymphatic vessels. Then the lymph node will filter out those pathogens, destroy those pathogens. Look at all the different immune cells that are found in a lymph node. And then the lymph drains out of the lymph node in efferent vessels. There's different regions of a lymph node. The outer cortex contains what's called germinal centers, which house a lot of our B cells. A lot of our B cells like to be in these outer germinal centers. Then when they get activated, they migrate to this medulla area, and then they spit out antibodies. So these B cells will start to make antibodies, but the best place to be would be right here because that's where the fluid is draining out. So now you got B cells, plasma cells, producing antibodies, antibodies coming out into the lymph, and then the lymph going into your blood. Now you got antibodies in your blood. And once you have antibodies in your blood, those antibodies can get almost everywhere. They can circulate almost to every tissue in the body. The only thing they can't do is get into a cell. Antibodies cannot get into cells. So if you've got a virus that's inside of a cell, you have to destroy the cell because antibodies can't help you. So antibodies get to anything that's outside of cells. Regional names to be familiar with. Axillary are the lymph nodes in your armpit. Cervical are the lymph nodes in your neck. Mediastinal are the ones in your mediastinum, your chest. Hilar has to do with the hilum of a lung. Those are really important when we're looking for lung cancer. We want to know whether that lung cancer has spread. And hilar lymph nodes are right where anything that comes in or out of the lung. It's kind of the entry and exit of a lung. It's called the hilum. So lymph drains out at the hilum. So we test those lymph nodes. Mesenteric are found in your intestines. Inguinal are in your groin. Femoral are in your thigh. These are all very common locations to find many of our lymph nodes. Other secondary lymphatic organs, very similar to lymph nodes in that they harbor and house a lot of our immune cells, a lot of our lymphocytes. The spleen, very large mass of lymphatic tissue, located in the left hypochondriac region of your abdominal pelvic cavity. So basically way upper left in your abdomen, between the stomach and diaphragm. Contains what's known as white pulp. That's the lymphatic tissue. And red pulp, which we've already learned what the red pulp does. Removes worn out red blood cells and platelets, stores platelets, and as a fetus, it's where blood cells are produced. But this white pulp is the new idea, in that you've got a lot of immune cells that can remove blood-borne pathogens. Any pathogen that has gotten into your bloodstream, we can use our spleen to get them. Other lymphatic tissue is called nodules. These are masses of lymphatic tissue that is not enclosed in a capsule. Lymph nodes have a capsule. The spleen has a capsule, which is kind of a shell, an outer membrane. These guys don't. A lot of them are found in your mucous membranes. They're called MALT, mucosa-associated lymphatic tissue. Your intestines have a ton of MALT because there's a lot of microbes that might try to penetrate when you ingest them. So you have a lot of lymphatic tissue in your intestines called MALT. Your tonsils, we have 5 tonsils, all strategically located where you either breathe in air or ingest food, because that's how microbes can get in. There's one at the back of your throat called the pharyngeal tonsil or adenoid. It's in an area of your throat called the nasopharynx. That's the uppermost part of your throat. It's at the back of your nasal cavity. So this is the guy you have to actually look through the nasal cavity to see. You can't see it in the oral cavity. The ones you see in the oral cavity are your palatine tonsils. There's 2 of those on each side of your soft palate. And they can swell like golf balls. And they can block airflow, and they can be problematic. And then lingual tonsils are at what we call the base of the tongue, way at the back of the tongue, are the 2 lingual tonsils. There's some tissue called Peyer's patches in your ileum. That's the last part of your small intestine. Again, just tissue that's loaded with immune cells. Next video, we're going to get into a concept called antigen presentation. So that's cells have the ability to present antigen, show antigen to other immune cells.